Hello everyone. We continue to study physiology of nerve system, and today we have practiced 7 structure of synapses and mediators function. As usual, we have a lot of links, a lot of video, and even on our page in the Moodle we have short video files. You see a neuron synapse, muscular system synapses, and neuromuscular junction. And let's start studying it from classification of synapses. And I hope you know, in a nerve system, a synapses is a structure that permits a neuron or nerve cell to pass an electrical or chemical signal to another neuron or to the target effector cell. Neuron synapses can be chemical or electrical, also the majority are chemical. Chemical synapses are the result of neurotransmitter realize and always travel in one direction. And you see the chemical mediator transmitter molecule can be excitatory, inhibitory, and modulatory. So, and synapses can be excitatory, inhibitory, and modulatory. Electrical synapses, they are only excitatory synapses, and sometimes in some books describe electrochemical synapses. And you see the part of the cell, neuron cell, has a lot of contacts with these places, and even green and red color. And you see here red color inhibitory synapses and green color excitatory synapses. And all synapses can contact with a cell in one time. So, next slide. Discuss about electrical synapses. They are only possible when two cells are connected by special channel proteins. The synapse can travel in both directions. This is rare. A very limited number of neurons can communicate by way of an electrical fluid without charged ions traveling through synaptic channel proteins. Connexon. Here you see a lot of connexon channels and even mechanical contacts through these proteins. Right here you can see them in microphoto. Even here, next slide, you see a lot of connexon channels on the surface of membrane, and here you see connexon channels through the B-phospholipid membrane. And small ions and little molecules can move through these channels. Here you see description. An electrical synapse is much quicker than a chemical synapse. It does not depend on the availability of neurotransmitters and does not need to wait for neurotransmitter vesicles to open or not the neurotransmitters to travel through the synaptic cleft or for the next cell to absorb and respond to these neurotransmitters. The neurons of the hypothalamus use electrical synapses to secrete hormones. Some neurons in the hippocampus communicate with this way with electrical fields. Difference between synapses on this slide in a table. You see difference between electrical and chemical synapses. You can find all these differences, especially in gaps between the cells. You see in a chemical synapse it is about 20 nanometers, and the smaller gaps only 3-5 nanometers. It's very, very close contact. So next one, cell attachment. We have looked at two different functional types of synapses, but synapses can also be classified according to how they connect to other neurons or other cells, and even location. We see a lot of different way to contact: axonal, axodendric, axomuscular, dendrodendric, somatic. The nature of excitatory and inhibitory and electric and chemical, you know it. Types of attachment between two cells include, so describe: axodendric, the axon of one neuron contact with dendrite, by far the most common synapses in the body. Axosomatic, the axon of one neuron connect to the cell membrane of the body or soma of another cell. Axonic, the axon of one neuron connect to the axon of another neuron. Usually these are inhibitory synapses. Dendrodendric, for example, these are dendrite connect between two different neurons. And neuromuscular synapse, the axon of one neuron connect to a muscle. You know these synapses from previous lesson, I hope. And neurons connect to all parts of the body. Various others include axons in the intestinal space, or to a blood vessel, for example. Here you see synapses can be classified on the type of neurotransmitters. Examples of neurotransmitters include dopamine, adrenaline, acetylcholine, or others. This help name the synapses according excitatory or inhibitory, and even except for acetylcholine. How I said, the excitatory transmitters and inhibitory transmitters can work together in one time. In this place, axon hillock is a place of decision making, where process integration, summation in this place, and the further transfer of information. And here we see another type of chemical synapses. These are modulating synapses. Modulating synapses by adrenaline synapses. Adrenaline molecule has mixed action. The action depends on which kind of receptor cells this mediator act on. It's time to learn to structure the synapse. You see the classic chemical synapse. In general, structure and arrangement is similar to that of the neuromuscular junction, and a great deal of information about central synapses has been derived from experiments on the point of contact between nerve and muscle cells. We will use this example to in the future we will watch the video. And what is the structure of a synapse? The synapse consists from three parts. The first, the presynaptic membrane axon terminal of the neuron that is sending information. Exactly, it can be not only axon, you know, dendrodendric synapse, it can be dendrite or axosomatic. This place can be axon, this soma of close neuron. Next, synaptic cleft. It's little spice, a teeny 20-30 nanometers wide gap between the two neurons filled with a fluid, call it the intertransmitter spice, interstitium liquid. The postsynaptic membrane, it's next part of the synapse of a second receiving cell. Is usually another neuron, but it might also be a gland, organ, or muscle. The postsynaptic membrane has protein channels. Here they are in the blue line. These protein channels call it receptors, and they are more abundant here than in other parts of the cells. In presynaptic is before the gap, synaptic cliff, and in postsynaptic is after the gap. So it's almost all, but here you see the neuromuscular synapse. Differences from interneuron now. The synapse, the spice, postsynaptic membrane is more active area of the postsynaptic membrane, more active zones. The mediator is only acetylcholine, and acetylcholine esterase break down this molecule. The receptor channel is ionotropic. In neurons, they are ionotropic and metabotropic G proteins. Here you see the work of these proteins, special proteins in a cell membrane. In neurons, they are ionotropic and metabotropic, bound to G protein channels that trigger a cascade of metabolic reactions that enhance the effect of the mediator without increasing its concentration. So we don't need so much transmitter molecule for transmit the signal. Let's look at the stages of synaptic transmission, chemical transfer across synapse. So stages of synaptic transmission. When an action potential reaches the axon terminal, it triggers the opening and voltage-gated calcium channels. At first, it's very important to have enough calcium ions. Here, it's very little ions, and so we look these ions move through the membrane, and here we see. These calcium ions diffuse into the cell and promote the fusion of vesicles containing neurotransmitter. Here we see little molecules of transmitter with the cell membrane. These little vesicles move to the presynaptic membrane. The neurotransmitters are realized from the axon terminal by exocytosis. Here we see. And cross the synaptic cleft, just move in a liquid. Here we see a lot of molecules of acetylcholine, for example, move through the cleft. Neurotransmitters bind to the specific receptors on the postsynaptic membrane and open ligand-gated ion channels, special channels you see, and opening of ion channels generate an electrical impulse in the postsynaptic neuron, propagating the presynaptic signal. The neurotransmitters realized into the synapse are either recycled by reuptake pumps or degraded by enzymatic activity. We will discuss about this next. And here you see all the stages and even more. The neurotransmitter, neuromuscular junction, like example. First, an action potential in the motor axon invades the nerve terminal and depolarizes the membrane. This depolarization open voltage gating calcium channels, and calcium ions enter the nerve terminal by diffusion down the electrochemical gradient. This leads to the rise of in intracellular free calcium, which triggers the fusion of docked synaptic vesicles with the plasma membrane, and acetylcholine contained within the synaptic vesicles is realized into the synaptic cleft. Acetylcholine molecules diffuse across the cleft and bind to the nicotinic receptors. This new word, nicotinic receptors, but in the part of nerve system, autonomic nerve system. We will study it soon. Next practice, we will discuss about all kinds of receptors to acetylcholine, noradrenaline, epinephrine, and this word we will hear. On the postsynaptic membrane, when the receptors bind acetylcholine, they cause non-selective cation channels to open, and this depolarizes the muscle membrane in the end plate region. This depolarization is called the end plate potential. Finally, when the potential has recharged transhood, the muscle membrane generates an action potential that propagates along the length of the fiber. This action potential triggers the contraction of the muscle fiber. You know it from previous practical. And what we have here, you see all the stages. And next one, chemical synapses are the most common synapses in the body. The chemical synapses use chemical messenger molecules to generate an electrical signal, and these messengers that are generated in the postsynaptic cell are called neurotransmitters molecules. They diffuse into the synaptic cleft to bind to the receptors, to open gates that allow ions to flow into the postsynaptic cell. Receptors are specialized protein channels that only allow positively or negatively charged ion move in the cell. So, in negatively ions, in this case we have chlorine molecule and not sodium chlorine ion. And next slide, you see all ways of mediators in activation. After work, this molecule of neurotransmitter should be inactivated. What way we can do this? At first, the enzymatic breakdown of transmitter. The second, repeat inactivation of cation channel. And one more, we have this part of postsynaptic membrane. We just move it inside. Internalization of receptor, and this protein can't contact with the neurotransmitter. One more, diffusion out of cleft. So molecule of neurotransmitter just can move, flow anywhere. You know that we have a nerve system a lot of glial cells, and these glial cells can just break down and this molecule of neurotransmitter can just extraneuronal uptake of glial cells. Reuptake, one more way, just this molecule move from special channel to the presynaptic part of the synapse. inhibition of exocytose, and one more autoreceptor. We have special part which contact with the neurotransmitter, and this process just do inhibition of synthesis of neurotransmitter. So we have a lot of way to inactivate of neurotransmitter molecules. All this, what we know about all these molecules. So, neurotransmitters are chemical messengers that are realized from a synaptic vesicle into the synaptic by neurons, messengers that allow nerve cells to communicate with each other. A neurotransmitter signal travels from a neuron across the synapse to the next neuron. There are over 50 molecules types of neurotransmitter. We know some of main classification are described below in a few categories. You see amino acids, peptides, biogenic amines, and especially acetylcholine. You know this molecule. About classification, we have one more slide about classification. A neurotransmitter can influence neurons in one of three ways. Can be excite, inhibit, or modulate them. Excitatory neurotransmitters, these types have an excitatory stimulating effect on the neurons. If a neurotransmitter is excitatory, it will increase the likelihood that the neuron will fire action potential. Examples of these types of neurotransmitter, the epinephrine and norepinephrine, adrenaline and noradrenaline. Inhibitory neurotransmitters, in contrast to excitatory neurotransmitters, inhibitory neurotransmitters have the opposite effect. Inhibition the neurons. If a neurotransmitter is inhibitory, it makes the likelihood of the neuron firing action potential will be decreased. Examples you see in these types of neurotransmitter and endorphins, lysine, serotonin, and modulatory neurotransmitters, both. These are often called neuromodulators. If a neurotransmitter is a neuromodulator, this means it can affect a large number of neurons at the same time, as well as being able to influence the effect of other neurotransmitters. Neuromodulators do but work together with neurotransmitters to enhance the excitatory or inhibitory responses of the receptors. Examples for these types of neurotransmitters are serotonin and dopamine. Norepinephrine especially will discuss about this kind of neurotransmitters in autonomic nerve system. With the neurotransmitters is excitatory or inhibitory is depend on the receptor. It binds to on the postsynaptic neuron. Some neurotransmitters can be both excitatory and inhibitory, and some can activate multiple receptors, as there is not just one receptor for each type of neurotransmitters. So we can find all these molecules here. These substances have different effects on our nerve system. And now examples of neurotransmitters, and some of them we will discuss more. For example, norepinephrine, or you hear adrenaline. Adrenaline is a primary hormone released by the adrenal gland, but some neurons may secrete it as a neurotransmitter and increase heart rate and blood flow, leading to a physiological boost and physical boost, and heightened awareness. It's produced during stressful or exciting situation. The next one, almost the same, norepinephrine. It contrast to adrenaline. Norepinephrine is predominantly a neurotransmitter that is occasionally realized as a hormone. It contracts blood vessel contact with the receptors in the blood vessel and increases blood flow, improving attention and the speed in which response action occur. And you see the slide shows the history of discover this molecule. Next one, dopamine. It's primarily responsible for feelings of pleasure, but it's also involved in movement and motivation, especially for humans. People tend to repeat behaviors that lead to the dopamine release, leading to addictions. Abnormal dopamine secretion is common in specific movement disorders like Parkinson's diseases. Even we have the drugs L-DOPA for treatment these diseases. And history of discovered these dopamine molecules. Serotonin contributes to feelings of well-being and happiness, is affected by expressive, regulates attention or other complexes, cognitive functions such as sleep, dreaming, eating, mood, pain regulation. And if you have too little serotonin, has been shown to lead to depression. And you see a little history and disorders with this molecule. Aminobutyric acid, this neurotransmitter with the inhibitory effect. It's most common neurotransmitter present in a high concentration in its central nervous system, especially in the brain, preventing the brain from becoming overexcited. Its GABA is lacking in central parts of the brain and results as epilepsy, and also contributes to motor control and vision. Next one, acetylcholine, involved in serotonin and memory within the brain, activates muscle contraction in the body, and it's also associated with the attention and a lot of function, you see, and and related to Alzheimer's diseases. If we lose this molecule and history, a lot of time, almost 100 years, we know this molecule. One more, glutamate, most common brain neurotransmitter, regulates development and creation of new nerve pathways, and hence is involved in learning and memory process. And what else? In addition to neurotransmitters, there are molecules similar in the function, these neuromodulators. In turn, they divided in four large groups: neuropeptides, and endorphins, methencaphalins, calcitonins, substance P. We have fatty acids, derivatives, and purines and pyrimidines, and even ATP molecular is a neuromodulator too. And we have the gaseous substances different. So, and more two mediators can work in a synapse. Here you see big, large vesicles, and usually we say that it is dark vesicles, which contains a mediator that has a large molecule, for example peptide. It does not connect with the active zone, is not realized at all at a low frequency in this stimulus. And we have small light vesicles with a mediator that has a small size molecule, and these vesicles can contact with the active zone of presynaptic membrane and a low frequency of the stimulus. And sometimes this helps to do the difference in the effect of stimulation. A mediator with large size molecular peptide molecular at a high frequency of stimulus is realized, but not in the active zone. In the active zone work mediator from light vesicles together, and it work in the same time, in the same place, but with a difference in the effect of stimulation. So it is almost all about synapses. We don't have lab works for today, we just have a lot of video. I will leave this video and links to this video in description to this practice, and don't forget to do your test in our Moodle page in our site. So see you later, bye bye.